All right, this is your fresh new video. If I can get it going. All right, this is actually chapter 12 of your textbook. The cell cycle is the life of a cell from origin through division. Some cells divide frequently, some do not. An example, your skin cells continually divide. Brain cells, not so much. The cell cycle consists of interphase and mitosis. So let's talk first about mitosis, actual cell division. Mitosis is technically the division of the nucleus, so all the genetic material. And cytokinesis is the division of the cytoplasm. This is what actually pinches off and makes those two separate cells. Mitosis plus cytokinesis is about 10% of the life of a cell. The phases of mitosis are prophase, metaphase, anaphase, telophase. And I was on this key math. So that's here. This is about 10%. The rest, about 90% of the life of a cell, is actually going to be an interphase. This is a phase of non-division. This is getting ready to divide, but not yet divided. Here's a picture of the different phases of mitosis. Here's prophase, where the nuclear membrane is breaking down, and the chromosomes have actually coiled up. You can't actually see chromosomes under the microscope until they're in a division phase. Otherwise, it just looks like a massive tangled DNA called chromatin. But once the cell enters into prophase, you can actually see them coiled up into these chromosomes, and you'll get to look at some from an onion root, this unit. And then in metaphase, all those chromosomes, once the nuclear membrane is broken down, all of those chromosomes are going to line up in the middle of the cell, and then they're going to split apart. Now, one chromosome here, if I'm just going to kind of pretend and blow it up here, is actually one chromosome attached to another chromosome, even though it looks like an X. And they're attached with a centromere in the center here. And when they're like this, we call it a chromosome, but it's actually two sister chromatids. And the reason it's called that is because this chromosome is an exact copy of that one. So back in interphase, we'll talk about how DNA makes a copy of itself. Well, when it does that, these copies attach to each other, and they're attached by a centromere. Now, when the cell divides, it's going to pull those sister chromatids apart. At that point, we just call them chromosomes. And then they're going to divide, they're going to split apart, and then a new nuclear membrane is going to redevelop in telophase, and then the cytoplasm is going to split. Cytokinesis, which is not technically part of mitosis, but cytokinesis actually begins during telophase. So why do we do this? Well, the big idea, which is what we don't want to lose here, is that mitosis allows cells to go from 4N to 2N. Well, there's not really a 4N. What that means is number of chromosomes. 4N kind of refers to what happens after a cell undergoes DNA replication. So, for example, in a human cell, there are 46 chromosomes. When that cell goes through interphase, typically the X phase, all that DNA is going to be copied. So theoretically, you could say there's like 92 or 4 for a brief moment. And then when those cells divide, that 92 gets split up into 46 and 46, so that these daughter cells look exactly the same as this original cell here, which is 2N. So this is where we get the 4N from. It's technically an interphase. For a brief moment, it is 4N. It produces somatic cells. Somatic is just a fancy word for body cells. And they're diploid. This is the algebraic expression for the term diploid that you will learn about in one of Bosen's videos. It does not produce gametes. Okay, those are just in. Does not produce gametes. What are gametes? Gametes are sex cells, like egg or sperm. Mitosis does not produce egg or sperm. It does not produce gametes. Mitosis produces body cells. Okay, anything that's not a reproductive cell. So a brain cell, a liver cell, an intestinal cell, a skin cell, a blood cell, anything that's not actually going to be put forth for reproduction. So we talked a little bit about this interphase, but what exactly is it? It's a phase in which the cell grows, it metabolizes, and this is really important, it copies the genetic material. This is what causes that chromosome to actually have two sister chromatids and be identical. This one is the same. I don't know how to indicate that. Same? Oh, I don't know. Interphase is about 90% of the cell cycle. So if you kind of go back, which apparently I can't do without hitting the back button here. Hang on. All right, sorry, I was interrupted there for a minute. So let's go back. And, okay, so interphase is at 90%. So this is interphase out here. Okay, about 90% of the half-life of a cell. I have to hit that button. It's actually split up into three smaller phases. Gap 1 is basically where the cell is just growing. Okay, this is where it's still diploid. And then synthesis phase, which is F, is about 35% of the entire interphase. So this is where it duplicates that DNA. Okay. So it's going to create that later. And then gap 2 is going to be just more growth. Particularly here, we're getting ready for division, preparing for division. And there's still 4 ends so that they can divide later. So here's just another view of that. Now, interestingly, there's a G0 as well. And this is where a cell that is not ever going to divide will actually exit the cell cycle and go into kind of a non-dividing eternal interphase. This would be, for example, like a brain cell. They don't really divide very much beyond a few years after birth, I think. I'd have to look up that statistic exactly how long those continue to develop. Your brain cells develop a lot in adolescence, but they develop. I don't think that they reproduce much after first year or two of life. And then these are just pictures of mitosis underneath the microscope in cells. So this is interphase. It looks just like a cell. And there's a clear nucleus right here and the DNA material, but you can't actually see, girls, but you can't actually see individual chromosomes. Prophase, the nuclear membrane is a little less visible here, and you can actually see the chromosomes that have coiled up. In metaphase, you can see these are spindle fibers that have attached to the chromosomes, and then all the chromosomes are right there in the middle. We call that the metaphase plate. And then in anaphase, they start to pull away from each other. This isn't a very good anaphase. This is probably a better anaphase, but they pull away from each other. Those sister chromatids are now pulling away. This would be early telophase, very late anaphase. You can start to see how the cell is going to divide right here. That's where it will eventually divide. And then by the end of late telophase, it's not only started to divide, it's gone through cytokinesis, but it's starting to reform that nuclear envelope right there. So, kind of summarizing what's actually happening in each of the phases of mitosis. Prophase, the chromatin, this is just uncoiled DNA, where you can't actually see any chromosomes. That's where it condenses into the chromosomes. So now, so you can see those chromosomes are visible right there. They're made of sister chromatids attached to the centromere. So here's a sister chromatid, here's another one, and there's going to be a centromere attaching them right there. This is also where the microtubules form. This is the spindle, or the spindle fibers. The centrioles are going to move to the poles. So that's what's going to be kind of on each end here. In plant cells, those don't exist. And then the spindle is going to attach like that to the end. And then in metaphase, the microtubules will attach, again, these are spindles, will attach to the connectors, I'm not sure how to pronounce that, of each sister chromatid. So that's basically the center of it. The chromosomes will line up along the metaphase plate, so you can see them lined up here. You can still see the spindles attached to them, and then the centriole area right here. And then in anaphase, they're going to move away from each other. So this just shows kind of the progression of how far they're going to move. They're going to keep moving, moving, moving. The connector is right here. It's part of what that centromere was at one time. And they're just going to keep pulling and pulling and pulling until they pull them all away. And this just shows kind of an animation of what that would look like. And then this is that spindle fiber, and it just attaches and it's going to pull it. And then telophase. The new nuclei are going to form, the spindles are going to disintegrate, and then that's when cytokinesis is going to begin. Cytokinesis is actually the division of the cytoplasm. In an animal cell, it's a cleavage furrow, basically a buckle right here. The cell is basically caving in on itself on each side until they split apart. In a plant cell, it's going to create a cell plate right here, and then that plate is going to develop further into a cell wall. So these are just pictures, again, of mitosis. This is in a plant cell. And so, so you've got prophase, then you've got prometaphase, so somewhere in between prophase but not quite yet metaphase. This even looks to me a little bit more like an interphase, maybe close to interphase here, because those chromosomes aren't as clear as they are there. And then they line up in the middle, they pull away, and then telophase, they're going to split, and that's going to become a new cell wall right there. All right, so let's talk a little bit about how cells, or how your body controls the cell cycle. We do it by checkpoints and regulatory proteins. So between G1 and S, remember this is still an interphase, the cell receives the go-ahead signal from the environment that tells it that it needs to go on to the S phase to duplicate its DNA. So what do we mean by signals? Well, think back to your cell communication. Okay? The cells are going to communicate with each other, are going to communicate with each other to send their signals to neighboring cells. Then the checks are enough mass in the condition of the DNA. Then there are checks for enough mass, so the cell's big enough and the DNA is good enough so it can divide. If there's no single signal, the cell is going to go into the G0. So if the cell doesn't receive the signal, it's going to go into a non-dividing stage. I talked about that before. So like things like brain cells would go right into that. Most cells in G0 never divide, nerve or muscle cells. Muscle cells also don't divide. When you get bigger muscles, you're actually enhancing those muscle cells. You're not actually making more of them. And they only divide if there's some kind of injury. And even brain cells don't necessarily always regrow. I mean, when you get a brain injury, a lot of times other parts of your brain take over. You don't really build new neurons. Same thing for drug use. Don't do drugs. So then between G2 and starting mitosis, it's going to check for, again, the mass, make sure there's correct DNA replication has happened in the S phase. Okay? If all is okay, the cell is commit and it's going to enter into the M phase and divide. Note, cancer often occurs because cell is quickly pushed from the G1 to S without the proper checks. This is a big thing with cancer, is that these checkpoints that are so important are not working properly. Often the result is a damaged P53 gene. We're going to talk about that in another set of notes later. But what it is a tumor suppressor gene. And it actually checks for DNA damage. And if the DNA is damaged, it will shunt that into like a G0 stage instead of letting it divide. Well, if this gene isn't working and it's not checking it, it will commit a cell to divide when it shouldn't divide because it's damaged. So that's where you get heavily damaged cells continuing to divide and making more heavily damaged cells. So here's my next page. Nope. I missed something? I did. Nope. Okay. We're getting the hang of using this as a tablet. Okay. So checkpoints, this is kind of showing you what's going to happen. So we've got a cancering mutation, something that's not right in the DNA. With a P53 cell, it will either repair it, or the cell will die, or it could go into G0, and everything's happy. maybe it's repaired or the cell dies, whatever the case is, it's not going to make more of them. Unfortunately, without that P53, the cell may go mutation crazy, make more mutations, and then make more copies of the cell that have the mutations, and then that leads to a tumor. Regulatory proteins or enzymes. Cyclin-dependent kinases are enzymes that are needed to drive the cell cycle, make it go forward. They work when activated by cyclin. Cyclin is a protein that rises and falls in the cell cycle. Example, maturation promoting factor MPF, better known as. It's a cyclin increase in concentration in G2 and binds with a specific CDK to form MPF. This signal is important because it starts mitosis, going back to cell communication and how that works. At the end of mitosis, the enzymes break down cyclin, so there's no MPF and no more dividing. And then the CDK concentration remains the same. Touch together. So here's another picture of kind of talking about that in terms of the cell cycle right here. New cyclin accumulates, then we have cyclin, makes MPF, then we go to a checkpoint, and then it's degraded. Other internal signals that it must have, the kinetochores must be attached before anaphase can occur because that's what it attaches to before it pulls them away. And the unattached kinetochores are the signal to stop the cyclic chromatids from splitting. So if there's a problem there, that's a way to stop the cell from finishing its division. Other things that can happen, external signals that come from the outside environment, growth factors, proteins that are released by certain cells that stimulate other cells to divide. Remember that cell communication piece. Cells are going to communicate with each other, and if those growth factors are too many or too little, that's going to affect how quickly or how not so quickly your cells divide. Density-dependent inhibition, crowded cells will stop dividing because there aren't enough growth factors and nutrients for it to divide. And anchorage-dependent, if cells aren't attached to the extracellular matrix, they will not get growth factors, so they won't divide. So there are normal ways that growth factors are used to either regulate or not regulate cell division. Okay, so then this is reproduction in prokaryotic cells, because technically this is similar to mitosis in multicellular organisms, like you. So bacteria, they don't really have chromosomes. They usually have one loop of DNA, a circular piece. Sometimes it's longer than that. And it kind of looks more jumbled like this. It's not really circular, but it is attached to both ends. Only one set of genes. They don't have a mom and a dad set because they're not eukaryotic and they don't reproduce sexually. They may contain one or more plasmids. A plasmid is like an extra loop of DNA that's smaller, and that DNA is able to move from one bacteria to another, which is kind of cool because that's how you get a lot of genetic variation in bacteria because they don't actually sexually reproduce. However, that can be used in engineering because we can take a gene that's really important to us and we can insert it into that plasmid, and then it will share it with all the neighboring bacteria. And we can genetically engineer them. We will actually do that in a bacterial transformation lab in probably January, February. So this is an example. This is good for bacteria because it allows for recombination. It allows for genetic variation without sexual reproduction. So it's asexual reproduction. The basic steps: DNA replication, the cell pinches off. There should be no genetic recombination unless they have a plasmid or there's some kind of mutation that happens to DNA, and it's really fast. That's one of the biggest advantages of asexual reproduction. About every 20 minutes, depending on the bacteria, you can get offspring. Budding, some prokaryotic organisms reproduce in this manner. Yeast also reproduce by budding. Basic steps, the cell develops a bulge or a bud, right there. The DNA copies, and then the bud breaks off and grows. And that's it.